Good morning, students. Hope you all are fine and safe. Today we are going to study Science 2. In that, we will study remaining topic of lesson number 2, Life Processes in Living Organism Part 1. Now let's study energy from different food components. Excess of carbohydrates are stored in liver and muscles in the form of glycogen. Proteins are the macromolecules formed by bonding together many amino acids. Proteins of animal origin are first class proteins. We get 4 kilocalorie of energy per gram of proteins. Amino acids are obtained after digestion of proteins. Those amino acids are absorbed in the body and transported up to each organ and cells via blood. From these amino acids, organs and cell produces various proteins necessary for themselves and the whole body. For example, melanin and keratin is produced in skin, osin is produced in bones, various proteins of cell membrane, various enzymes are produced in cells, insulin and trypsin is produced in pancreas, various hormones are produced in pituitary glands, actin and myosin, flexible protein, is produced in muscles, hemoglobin and antibodies are produced in blood, etc. Students, can you recall from where do we obtain the lipids? The substance formed by a specific chemical bond between fatty acids and alcohol are called as lipids. Digestion of lipids consumed by us is nothing but their conversion into fatty acids and alcohol. Fatty acids are absorbed up and distributed everywhere within the body. From those fatty acids, different cells produces various substances necessary to themselves. Example, the molecules called as phospholipids, which are essential for producing plasma membrane, are formed from fatty acids. Besides, fatty acids are used for producing hormones like progesterone, estrogen, testosterone, aldosterone, etc. and the covering around the axons of nerve cells. We get 9 kilocalorie of energy per gram of lipids. Excess of lipids are stored in adipose connective tissue in the body. Students, do you know what are vitamins? Vitamins are group of heterogeneous compounds of which each is essential for proper operation of various processes in the body. There are main 6 types of vitamins. Example, A, B, C, D, E and K. Out of these, A, D, E and K are fat soluble, whereas B and C are water soluble. We have seen that FADH2 and NADH2 are produced in the process like glycolysis and Krebs cycle. Vitamins like riboflavin, that is vitamin B2, and nicotinamide, that is vitamin B5, respectively are necessary for their production. There is about 65 to 70% water in our body. Each cell contains 70% water by its weight. Blood plasma also contains 90% of the water. Functioning of cells and thereby whole body disturbs even if there is a little loss of water from the body. Hence, water is an essential nutrient along with all above mentioned nutrients. Fiber are also essential nutrients. In fact, we cannot digest the fiber, however, they help in the digestion of other substances and ejection of the undigested substances. We obtain the fiber from leafy vegetable, fruits, cereals, etc. Now let's study cell division and essential life process. Cell division is one of the very important properties of cell and living organism. Due to this property, only a new organism is formed from existing one. A multicellular organism grows up and emaciated body can be restored. There are two types of cell division as mitosis and meiosis. Mitosis occurs in somatic cells and stem cells of the body, whereas meiosis occurs in germ cells, that is reproductive cells, sperm in testes and ovules in ovaries. Before study of cell division, we should know that structural organization of cells that we have studied earlier. Each cell has a cell membrane as the outer covering of the cell. The jelly-like matrix in the cell is called as cytoplasm. It consists of nucleus which is surrounded by nuclear membrane. It has a perpendicularly arranged centriole in the cytoplasm. It has nucleolus present in the nucleus. DNA is made up of nucleolus which condense and form chromosomes. For example, if we considered a needle and thread, when very fine thread is coiled, the thread is formed, and when the thread is coiled, the reel is formed. Similarly, DNA is coiled to form chromatin fibers, and chromatin fibers are condensed to form chromosome. DNA present in the nucleus form chromatin fibers, and chromatin fibers condense to form chromosome. This chromosome consists of two arms with centromere in the center. Two similar types of chromosome forms homologous pair, and it replicate to form similar chromosome, that is sister chromatids. In one human cell there are 46 chromosomes, so there is 23 pairs of chromosome. Diploid chromosomes are known as 2n. Before the cell divides, the chromosome always duplicates and parent cells form daughter cells. Now let's study mitosis. Mitosis takes place in two stages, that is chirokinesis and cytokinesis. Chirokinesis is nuclear division which divides the nucleus in two, and cytokinesis is cytoplasmic division which divides the cytoplasm of the cell. Chirokinesis takes place in four stages, that is prophase, metaphase, anaphase and telophase. Now let's study mitosis in detail. So students, mitosis is a process of cell division that makes possible regeneration of a body parts. To elaborate, mitosis is the separation of the nuclear chromosome into two identical daughter nuclei. It is divided into four pre-phases, that is prophase, metaphase, anaphase and telophase, and additional phase called as cytokinesis occur after complete nuclear division. Before a cell divides, enter mitosis is undergoes period of growth and rest. This period is called as interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condense and become shortened, thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with the pair of centriole, moves apart to the opposite pole, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosome to move along the middle of the cell, above the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structure called kinetochore at the surface. The kinetochore holds the chromosome and attaches them to the spindle. Anaphase. During anaphase, the sister chromatid separates at the kinetochore and The daughter chromosomes, along with the divided centromere, move towards the opposite pole as the microtubule shorten. The poles of the tubules also move apart and help to separate the chromosome. As the chromosome separates completely into two sets, one at each pole, anaphase is considered to be complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei form along the nucleoli. This triggers the reformation of nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animal cells, a cleavage furrow appears in the plasma membrane which decondense and divide the cytoplasm into daughter cells. Each contain one nucleus. In plants, new cell wall formation is figured at the center of the cell by the formation of cell plate. The cell plate grows outward. It means the existing wall results in two daughter cells. Now let's study meiosis. Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms. During the production of reproductive cell called gametes, meiosis is the reproductive division that reduces the chromosome numbers of a cell. Cell divides and redivides to form four daughter cells. Student, always remember, meiosis involves a single rounded DNA. Replication followed by two cell division, that is meiosis 1 and meiosis 2. The entire process of meiosis in the formation of four haploid daughter cells is half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome numbers get restored. Students, always remember, meiosis 1 include four stages, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Let's know about homologous before studying about the prophase 1. Each diploid cell has two copies of chromosome, one inherited from the male parent and the other from the female parent. These two copies come from different parents, are similar to each other and are called homologous. Now, let us look at the different stages of prophase 1 in detail. To begin with, let us learn about liptotin. The chromosome condense and become visible. The zygotine phase is followed by pecatine. During pecatine, the chromosomal crossing over takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination module to be visible. As crossing over completes, it marks at the end of the packaging phases. Diplotin is followed by dikinesis, which is also the last stage of prophase 1. During dikinesis, the chromosomes are fully condensed and the nucleolus and nuclear membrane dissolve and the meiotic spindle begins to resemble. Prophase 1 is followed by the metaphase 1. During metaphase 1, the microtubules from the opposite poles are attached to the kinetochores and the bivalent align around the equatorial plate with a random orientation. Either of the parent homologue takes up on the any side of the spindle. The metaphase 1 is followed by anaphase 1. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of microtubules. The sister chromatids on the contrary remains attached to the centromere. Now let's study meiosis part 2. Students, in this session of meiosis part 1, we have learned the process of chromosomal recombination. Let us recall that the process of mitosis 1, in prophase 1, the chromosome link together to form a tetrad shape. Crossing over of gene occur. In metaphase 1, the spindle attached to the centromere of homologous chromosomes of each tetrad. In anaphase 1, two chromosomes of each tetrad separate and starts moving towards opposite poles of the cell. In telophase 1, the homologous pair reach the poles of the cells. A nuclear envelope forms around each chromosome, set the spindle disappear and cytokinesis follows. After cytokinesis, each of the two daughter cells has a nucleus with a haploid set of replicated chromosomes. The two cells that are formed in meiosis 1 undergoes similar stages in the next process and therefore called meiosis 2. In meiosis 2, the amount of DNA, that is the chromosome number, is reduced back to normal by splitting the chromosomes. Meiosis 2 is also divided into 4 stages. They are prophase 2, metaphase 2, anaphase 2, telophase 2. In prophase 2, the centriole divides into two and each centriole migrates to the opposite pole. The nuclear membrane disappears and chromosomes with two chromatids become short and thick. During metaphase 2, the chromosome arranged on equator or spindle, the centromere divides into two and each chromosome produces two chromatids. The spindle attached to the centromere of the chromosomes. In anaphase 2, the spindle fiber contract and chromatids move towards opposite poles. In telophase 2, the chromatids completely migrate to the opposite poles and are known as chromosomes. A nuclear envelope reforms around each set of chromosomes. Cytokinesis occurs, producing 4 daughter cells, each with a haploid set of chromosomes. So students, what is meiosis? Meiosis is a nuclear division occurring in reproductive organs resulting in gametes formation. Meiosis has two distinct divisions, that is meiosis 1 and meiosis 2. In meiosis 1, chromosomes are still diploid but with genetic exchange. In meiosis 2, the two cells divide into 4 haploid cells, each with half number of chromosomes. Hope you have understood the topic. Thank you. Stay home, stay safe.